Hi, I'm Brad Roth. I'm going to talk to you about a 2-semester intermediate course sequence in physics for the life sciences. So this is 2 semesters. We teach biological physics in the fall. We teach medical physics in the spring. A lot of you are interested in introductory physics. This is an intermediate class. I assume it's a prerequisite that you've had a basic introductory physics class. Also, we use calculus. So I assume you've had at least 2 semesters of introductory calculus. Ideally, these introductory physics and calculus classes would be at the university level, but I think a really good high school physics and calculus classes would be sufficient. What sort of students take this class? Well, we get physics students, physics majors. We have a medical physics undergraduate degree at Oakland University where I work, so we get medical physics majors. And we also get the biomedical engineers. Well, before Oakland University started its biomedical engineering program, we offered these classes once every 2 years and had like maybe 6 students in them. It was a struggle to get enough students to justify offering the class. I managed to get the 2 classes incorporated into the biomedical engineering curriculum. And now we have each class is offered once a year, and we have maybe 20 students each semester. So getting the biomedical engineers involved was crucial to the success of these classes. Occasionally I'll get a biology major, occasionally a chemistry major. I really try to recruit pre-med students, but the last thing they want to take is another semester or 2 of physics. So I haven't been very successful in getting the pre-meds. Now I teach this class as basically a mathematical modeling workshop to try to use mathematical models to show how physics applies to medicine and biology. That's the goal. The textbook I use is Intermediate Physics for Medicine Biology, which is by Russ Hobby and myself. So Russ Hobby was the author of the first 3 editions of this book. I became a co-author on editions 4 and the current edition, edition 5. If you want to learn more about the book, you can look at the book website that's given here. The book also has a blog. The blog gets updated every week. It has useful information for instructors, sample syllabi, new homework problems, background information from topics we just couldn't fit into the book. You might be interested in the book blog if you're interested in physics applied to medicine and biology. Okay, let's talk about what's in these 2 classes. First, biological physics in the fall. The first day, I like to talk about distances and sizes. I found that biology majors often have a good idea of the cellular structure, what cells look like, what bacteria look like, what viruses are, but what they don't have is a good idea of the relative sizes of all these objects. So we talk about relative sizes right from day 1. And then we go into biomechanics. I like to have a sample problem that sort of serves as a central idea of each lecture. And the central idea I use for the biomechanics day is lifting with your back. So what we do is we treat the spine as a rigid rod. So it's a simple model of what's actually a complex phenomena. As you lift something, your force gets transmitted through your shoulders up into the top of the spine. Your spine presses on the pelvis, so you get a reaction force R. And you also have this force F, which is the back muscle that's operating on the spine at a very shallow angle. Now, what you can solve this problem just using regular statics. And when you solve it, you can find the force on the back F as a function of this angle theta. Now, the key is to make sure the students, once they solve the problem, understand what it's telling you physically. That is, if theta is really small, so you're leaning over with your back like this and lifting, what you find is the forces are enormous on the back muscle. However, if you're lifting while staying upright, the forces are essentially 0 on the back muscle. So I like all my problems, all the classes to have a moral. And the moral of this story is that you lift with your legs, not with your back. So then we move on from biomechanics to fluid dynamics. And fluid dynamics is one of these topics that you just don't see in introductory physics, but so important in biology. And the problem I like to look at for fluid dynamics is viscous flow through a tube, where we can calculate the velocity profile. We can calculate the entire flow down a tube as a function of the radius, and it goes as the 4th power of the radius. So it's very sensitive to the radius of the tube. And this leads us naturally into the discussion of the circulatory system. Then we start talking about the exponential function. I tell my students that there's only one differential equation that I want them to know. It's dy dt equals by, the equation giving you exponential growth. And of course, if you have a minus sign in front of b, then you get exponential decay. And this comes up over and over through the whole semester. So we like to cover it right from the very beginning. For instance, you can put a constant source term in here. So you have dy dt equals a minus by, and you now have a nice model for infusion of a drug through an IV tube, and when the drug's cleared from the body by the kidney. So there's a lot of applications of this exponential function that the students really need to be comfortable with. And then this leads us to talking about scaling. Oftentimes scaling relationships are shown on log plots, so that brings in the connection with the exponential. Different animals of different sizes have to behave differently. For instance, as you increase the metabolic rate, it increases with body mass. But if you look at the specific metabolic rate, that is the metabolic rate per unit mass, it actually decreases. So a mouse, 1 gram of mouse tissue is very active metabolism, 1 gram of elephant tissue is a very sluggish metabolism. And those are those are scaling relationships, and different animals of different sizes just have to behave in these different ways because of some of the physics involved. Then we move on to biothermodynamics. And I like to look at heat capacity, and the problem I use is the dolphin. Dolphins are warm-blooded mammals that breathe air, okay? But they've evolved and adapted to live back into the ocean. So why haven't the dolphins evolved gills so they can extract air from the ocean like fish do, so they don't have to go up to the surface and breathe air all the time? Well, we look at this problem. We look at how much heat is lost because when blood is in contact with the surrounding water because of the in the gills, it's in thermal contact, and how much heat is lost to the surrounding water. So then you have to reheat the blood, and how much energy is used to do that versus how much energy do you get from the oxygen that you extract and you burn in metabolism. And the bottom line is it's a losing proposition. You lose more energy from reheating the blood than you get from the metabolism. So the dolphin's an example of how you can't have a warm-blooded fish, basically. You can't have a warm-blooded animal that uses gills. And it shows you some of the limits of what evolution can do. Evolution is a very powerful mechanism in biology, but physics provides limits to what can be accomplished. Then we go on to the heat equation. Yes, this is a partial differential equation. We do look at partial differential equations, but we don't go into them very deep. Basically, we might say, look at a steady state heat equation, and when we solve it, we guess a solution with some unknown coefficients, plug it back in, and try to figure out what those coefficients are that satisfy the equation, perhaps satisfy some boundary conditions. So kind of guess and check. So we are dealing with some advanced math concepts, but we're not doing it in a really advanced way. Now, the interesting thing about the heat equation is it looks just like the diffusion equation. So the next problem we solve is diffusion. The problem I like to solve when we're talking about diffusion is how big can a cell be? So a cell is undergoing metabolism. It's getting its oxygen from the surrounding medium. And in what point does the cell get so large that oxygen can't get transmitted into its center? And we can solve this problem. You can put in typical numbers for oxygen diffusion for metabolic rate, and you can find that the cells can get up to be about 1/10 of a millimeter in size, and you can't get any bigger. And if you want to get larger cells, or if you want to get a multicellular organism that's larger, you really have to develop a circulatory system to move oxygen around by convection instead of diffusion. So diffusion is another one of those topics that you just don't see in introductory physics, but is really crucial for understanding biology. Then we look at osmotic pressure. You have flow through semi-permeable membranes. So some molecules can go through, but some larger molecules can't pass through the tiny pores. And that brings us to the topic of the artificial kidney. I like to talk about dialysis, the artificial kidney, because all those biomedical engineers who are taking the class, they're interested in a lot of technological applications of these ideas. So it's a very nice chance to take our ideas of osmotic pressure, semi-permeable membranes, and apply them to an important biomedical process, the artificial kidney. Then we start talking about bioelectricity, and we have a fairly long section on bioelectricity. We study nerves and how they work, and that leads us to the cable equation, governs the nerve, the electrical potential along the nerve. Now, if you ignore that center term, the cable equation is just the diffusion equation, but it has that center term, so it gives a somewhat richer behavior than simply diffusion. You can set various terms to zero, solve it in special cases, and try to get a lot of information about how nerves work electrically. Also, you can make the membrane to be not a constant resistance, but to have ion channels open and close, and that leads to the Hodgkin-Huxley model. But that gets a little more advanced mathematically, and you really can't do much with simple analytical examples. But we do talk about the Hodgkin-Huxley model qualitatively. That leads us to what electric fields are produced by the body. And this brings us to the electrocardiogram. That is the electric potential on the surface of the body produced by the heart. And you can get the electrocardiogram is characterized by the P wave, the QRS complex, and the T wave. And you can understand the workings of the heart based on the electrocardiogram. You can also understand abnormalities and illnesses of the heart based on the electrocardiogram. And to try to treat these illnesses, we then look at pacemakers and defibrillators. Again, the biomedical engineers really like to learn about the pacemakers because they're the canonical example of how you can, of the medical device industry, how you can use physics and engineering to attack a medical problem. Then we have a chapter on biomagnetism. The biomagnetic effects aren't real big. You may want to skip this, but I spent my graduate student days measuring magnetic fields of nerve axons, so I'm really kind of hooked on biomagnetism. Shown here is a magnetotactic bacteria. There's tiny little iron oxide particles inside the bacteria that allow it to align with the Earth's magnetic field. And finally, one of the most important topics in physiology is feedback loops. So we spend a lot of time talking about various feedback loops. Feedback loops control the rate of breathing, the rate of heartbeat, blood pressure, glucose in the blood, all kinds of physiological parameters are controlled by these feedback loops. So we spend time studying them. Okay, so that was biological physics. Now in the spring is medical physics, and this is more physics applied to the hospital setting, clinical medicine. And we begin with sound, and this leads us to another partial differential equation, the wave equation. And we use it to look at the relationship between wavelength, frequency, we look at attenuation, we look at decibel scale, and we look at a little bit about how the ear works. And then we go on and we talk about ultrasound, using high frequency sound in order to do medical imaging. So this is a very common type of medical imaging that's done in hospitals. So we study ultrasound. Next, we look at light, and we look at the entire electromagnetic spectrum, not just visible light. We also look at ultraviolet light, which has a photon size that's large enough to actually break bonds, so you could potentially be causing cancer. And we also look at infrared light, which doesn't have a very large photon size, but can penetrate into tissue farther. So you can use that to image deep inside the human body, but there's a lot of light scattering. So there's a lot of interesting problems dealing with light. And then we look at the eye. And a couple of the problems I like to look at with the eye is, first of all, how you can treat nearsightedness and farsightedness using lenses. And also, I like to look at color vision. Color vision has fascinated physicists for years, going all the way back to Maxwell, studied color vision. Then the class takes a step up in its mathematical difficulty. We're going to talk about medical imaging, and to really understand medical imaging, you have to understand some about Fourier analysis. So we spend a few days in class talking about some simply mathematics of Fourier series, Fourier transforms. Once we have that, though, what can we do? We can look at tomography. That is, you can look at projections. You shine X-rays through a body object, and they are attenuated by the attenuation coefficient in the body along a line that you then record. And you can do this at different angles, and you get a whole series of projections. And you want to use those projections to understand what the attenuation coefficient is as a function of position in the body. That's the problem of tomography. And we can solve it in a couple ways. You can use filtered back projections, and then there's a total of Fourier method. Even the filtered back projection, by filtering, you're using Fourier ideas. So you see, Fourier analysis was really crucial if you want to understand tomography, and tomography is so important in the medical field. So then we move on and we talk about X-rays, how they interact with the body. You have the photoelectric effect, Compton scattering, paraproduction. We talk about X-rays, and then we talk about how you can use X-rays to do medical imaging. So here's an example of where you can take a medical image of the brain, then give it a contrast agent and take a medical image of the brain, subtract the two, and you get these beautiful images of the vascular tree that you couldn't see otherwise. So we talk a lot about X-ray imaging, and then we move on to nuclear medicine. Okay, we talk about some of the traditional topics of nuclear physics. We talk about isotopes. We talk about different mechanisms for radioactive decay, alpha, beta, gamma. But we also talk about some of the topics that are important in nuclear medicine. For instance, technetium-99m is a very important isotope in medicine. It's a excited state of technetium-99 has about a 6-hour, very long half-life for the excited state. And when it decays by emitting a gamma radiation. And that gamma is just perfect for doing medical imaging. So technetium-99m is one of the topics that we discuss a lot when we're talking about nuclear medicine. We also discuss about radiation safety, radon gas, these kind of things. Finally, the last chapter is on magnetic resonance imaging. We study magnetic resonance imaging and how you can make these beautiful images that we all are familiar with used in the medical field. Okay, so let me summarize here a few things about these classes. If you compare them with the typical biology major undergraduate classes, they have lots of math. But if you compare them by a physics major's undergraduate classes, not so much. Compared to, say, a electricity and magnetism class or a mechanics class or a thermodynamics class, the actual amount of math that we do is rather minor, modest. So, so I try to keep the level sort of in between what physicists and biology majors would expect. But the focus is on mathematical modeling. And if you're going to study mathematical modeling at a simple level, you really have to be dealing with toy models, spherical cells, and things like that. You have to have very simple models that you can solve analytically. Now, one thing I've found with students is sometimes they come into the class and they have good math skills and they have familiar with the physics and biology concepts. What they can't do is they can't interpret from the math to the physics, to the biology. That's one of the topics that I really try to hammer home. I want them to think. I want them to think before they calculate, after they calculate. I want them to think about how you translate from math to the physics and the mechanisms that implies. So we do a lot of drill and a lot of talking about taking mathematical equations, looking at special cases, trying to get the information out of the mathematics so that we can understand the real insights into biology that these mathematical models give. Now, we do skip parts of physics that don't have much biological relevance. We do almost nothing on planetary motion, Kepler's laws. We don't do much rotational motion. We don't do much high energy physics. Large parts of physics we just don't even cover because we want to focus on those parts that are of interest in medicine and biology. And finally, we emphasize technology. We emphasize artificial kidney, pacemakers, magnetic resonance imaging. These are topics that these biomedical engineering students are very interested in. So how physics can apply to different technologies. Okay, well, so that's the 2-semester intermediate course sequence I'd like to describe. If you have questions, you can contact me at roth@edu. If you want to know more about the book or the book blog, here are the websites. Thank you very much. Now, I'd be happy to answer any questions.